My name is Jennifer Bohannon and I'll be introducing Mycobacteria. Okay, so Mycobacterium is a genus within the Actinobacteria phylum. So there are 30 major bacterial phyla, and 7 of these are major pathogenic phyla. They include Actinobacteria, Firmicutes, Fusobacteria, Spirochaetes, Bacteroidetes, Chlamydia, and Proteobacteria. This is just an image of Mycobacterium tuberculosis in culture. This is just a type of Mycobacterium. Okay, so these are the 7 major pathogenic bacterial phyla. So we have the Actinobacteria, Firmicutes, Fusobacterium, Spirochaetes, Bacteroidetes, Chlamydia, and the big group, which is Proteobacteria. Alright, so the Actinobacteria phylum includes different genera of Mycobacteria, but some familiar ones include Mycobacterium tuberculosis, which causes TB, and Mycobacterium leprae. So yes, we can see that one here and over here. Yes. Alright, so this is just a key indicating important features of Mycobacterium tuberculosis. One is it's an emerging infectious disease. Another one, it's a CDC notifiable agent. It's also a globally important human pathogen. It's a validated biocrime agent. And it's also an NIAID Category C priority pathogen. So this NIAID, that's a categorizing system for emerging infectious diseases. Category C is actually the lowest priority, A is the highest. And just to give you a definition, an emerging infectious disease is a disease that has an increased incidence in humans over the past 20 years or threatens to increase in the near future. Okay, so to explain some features of the different types of bacterial cell walls, we have the gram-positive bacterial cell, which has the thick peptidoglycan layer. It also has lipoteichoic acid, which goes all the way through, and then teichoic acid, which goes partially through. We also have the gram-negative cell, which is a double membrane bacterial cell. It also has a thin layer of peptidoglycan here, and also has LPS in the outer membrane, sticking out from the outer membrane. So the gram-positive and gram-negative bacteria, they can be visualized using gram stains. So a gram-positive bacterial cell is going to stain purple, and then a gram-negative bacterial cell will stain pink. But mycobacteria, they can't be stained using a gram-positive or gram-negative stain, or using gram stains, because they have a waxy outer layer called mycolic acid. So you actually have to use a different type of stain altogether, and it's called carbol fusion. It's a basic dye. So that's the only thing that can pigment these waxy mycolic acid components out here in. It's visualized right here in red. But to give you some more detail about Mycobacteria, the cell wall structure, you first have a moderately thick layer of peptidoglycan, not quite as thick as this, but it is thicker than the gram-negative layer. Then next you have arabinogalactan, and then fused to that you have mycolic acid, which is just a fatty, waxy. These are fatty, waxy molecules here in red. Mycolic acid, yes. And then we also have glycolipids over here and polypeptides. So a familiar polypeptide is PPD, and PPD stands for purified protein derivative. It's also called tuberculin. So that's what's used in the tuberculin skin test to test for infection of tuberculosis, to test for tuberculosis infection. Okay, so we're going to talk now a bit about the structure and metabolism of Mycobacteria. So M. tuberculosis is a rod-shaped bacterial cell. It's also known as bacillus. It's also aerobic. It metabolizes glucose and triacylglycerides extracellularly, and it switches to a lipid-only metabolism intracellularly within macrophages. So once it invades macrophages, it switches to lipid-only metabolism. So this just gives you some pictures of the M. tuberculosis. This is a skin electron micrograph. You can see the bacillus shape here. Also down here, this shows the acid-fast staining of the Bacillus M. tuberculosis. You know, it's in red here. Background's blue. Then over here you can see the macrophages which have been invaded by M. tuberculosis in red. All right, so now we're going to talk about the steps of Mycobacterium tuberculosis infection. So the first thing is that it's transmitted through aerosols. So it's inhaled deep into the lungs, and once that happens, it will get lodged into the alveoli. So there are resident alveolar macrophages, and they engulf the M. tuberculosis bacteria. So the M. tuberculosis, it actually blocks the fusion of the lysosome to the phagosome, and that, by blocking it, it prevents its own digestion. So in that way, the M. tuberculosis is able to survive within macrophages. So it creates this intracellular niche, and it's able to replicate in there. It also can form these granulomas, and granulomas are multicellular structures that sequester bacteria from surrounding tissue. So once they form, they can actually become necrotic inside and also hypoxic. So sometimes granulomas will be able to kill the M. tuberculosis, but sometimes it just makes the M. tuberculosis enter into a dormant state. And you can live, some years like that. But the thing is that if a person becomes immune compromised, this could be because of AIDS or aging, as an example, the M. tuberculosis can actually become active again, and then it'll further infect the lungs, which causes bronchopneumonia. Yes, let's see where we are. Okay. So, so once it does that, can also not just infect the lungs, but it can actually spread to the lymphatic vessels and the vascular system. So once it does this, it'll affect other organs like the kidneys, the meninges in the brain, the adrenal glands, the liver, lumbar vertebrae, and also the cervical lymph nodes. Death from TB infection usually occurs because the lungs fill with blood and fluids. This results in hypoxia, respiratory failure, and suffocation. So over here we can just see the image of a normal lung on X-ray, and this is an X-ray of a lung with advanced TB infection. And so the white triangles are just showing you where the infected area is. It just highlights it right here. All right, so these are my citations here, and then my other group members will be talking a little bit more, giving more details about Mycobacteria.